global precipitation amount increased hugely within the last couple of decades, and we have receiving more rain in Turkey as well. According to the Turkish Meteorological Office, we have received the most rain in last year within the last 30 years period, and this huge amount of rain causing flash floods in the systems. And we wanted to test the impact of these flooding events in lake ecosystems using our mesocosms. What we want to test is that how much organic matter is coming from the catchment through the flooding events and changing the ecosystem structure and the functions through affecting the microbial and planktonic communities.
We are interested in the microbial communities and their response to dissolved organic carbon due to the increased flood events. To understand the community composition of microbial community, we will apply environmental DNA investigation technique. We collect our water samples and we filter these samples, and then with the next generation sequencing tools, we will understand the operational taxonomic units. And this will give us the compositional change, diversity change under the influence of this disturbance.
To test this, we had a pulse treatment of this dissolved organic carbon, and we want to follow this, how the system reacts to it. And we have two hypotheses. One is that if the systems are adapted to this kind of flooding events, they should resist the flooding and recover faster. And the second hypothesis is that if the systems are not adapted to, plus if the systems are less species rich in terms of the microbial community, phytoplankton community and zooplankton community, they will not resist enough the flooding events and the recovery will be slower. That's why we wanted to do this pulse treatment of the organic matter to the mesocosms twice. We did the first pulse and following everything in the systems with a different DOC, different color and different nutrients, and then 2 weeks after we will do the second pulse of the DOC, like the organic matter coming from the catchment, because we want to see that whether our system is learning and there is a sort of change in their resistance and the resilience of the system.
So from different DOC treatments, we expect the food quality for zooplankton community to change, and we want to relate these variations in the food quality, how this affects the zooplankton community. So based on these different treatments, we expect the food quality to change and to have a repercussion to the zooplankton community, and we want to measure that.
Today we're in the lab. So before you saw the mesocosms, those were big 2,000-liter tanks in the lake. Here we're doing parallel experiments to explain what's happening in those tanks, who's eating who basically, and how much. So to do that, we have these microcosms. They're usually half a liter, 1 liter, and we control the conditions. We add a certain amount of food and we add a certain kind of grazer. There's different kinds of zooplankton and each of them behave differently. So we want to quantify the effect of each kind of grazer to be able to explain what's happening in the mesocosm.
And through this, we will be able to say that how the flooding events are affecting the structure of the lakes and the function of the lakes, like in terms of carbon fixation, because carbon fixation is important for the global climate change. Based on these results, maybe we could suggest some mitigation measures, like how the catchments should be managed to stop the flooding events, even if the flash rains precipitation amount increase in the future.